Hi, I'm Alaf Hussain. Today I'm doing my project on the type 1 collagen protein. My mutation is a dominant negative substitution of glycine for a larger amino acid in several places. As you can see here, we have a 3D model of the original type 1 collagen protein. Right here, we have the mutant 3D model of the type 1 collagen. And as I place these side by side, you can see that the triple helix occurs at all locations on this original one. In contrast, in this one, there happens to be triple helix at only certain locations, and this is due to the substitution of glycine for another amino acid. And I'll get into details as why this occurs. First, we're going to start and tell you a bit background information of what type 1 collagen is. The type 1 collagen proteins are known as collagens in the matrix of the bone and cartilage. This protein is known to add structure and strength to the connective tissues and is found throughout the body. For example, in your skin, your tendon, your cartilage, your ligaments, and even between cells and tissues. This protein will be categorized as a structural protein because they are generally fibrous and stringy. Like structural proteins, type 1 collagen's main function is to provide mechanical support. Type 1 collagen is normally found in connective tissues including the skin, the tendon, and the bone tissue, as I mentioned earlier. This protein is also found between the spaces in cells and tissues called the extracellular matrix. Type 1 collagen is the single most abundant protein found in the animal kingdom. So normally you'll find them in animal cells and organisms. This protein gains a new function when mutation occurs. The mutation that occurs in the type 1 collagen protein is referred as to a dominant negative substitution of glycine. This mutation disrupts the triple helix structure, leading to a change in the type 1 collagen. As you can see here, you have your triple helix all around. As you can see here, it was disrupted. This mutation normally occurs in the collagen A1 or collagen A2 genes, which encode proteins involved in the formation of type 1 collagen. The mature collagen contains a triple helix region, one where you have a gene from the collagen A1 and collagen A2 that form this triple helix. Mutation normally occurs in the triple helix in either of the two genes that code for the type 1 collagen, as I mentioned earlier. Typically, in this triple helix region in the chain, glycine happens to occur at every third amino acid position. The glycine substitution in the collagen helix eventually hinders the formation of the triple helix. As you can see here, it's not a triple helix anymore, it's just 3 amino acid side chains that are connected. And you can see here that it's triple helix all around. Glycine is a single amino acid that occurs at every third spot in the amino acid chain of the collagen gene, which are involved in the formation of this collagen, type 1 collagen. But because glycine is small, it allows the alpha chains to form what is called a tight twist, or we refer to as a triple helix. This structural arrangement wouldn't be possible if an amino acid with a bulkier side, meaning it's larger than glycine chain, were substituted for glycine, which is exactly what occurs in the mutant. Essentially, the variations in coding cause a different amino acid to be substituted for glycine. This essentially alters the ability for the alpha chains to associate correctly when forming the triple helix. This abnormal protein is eventually hydrolyzed by the cell and simply fails to function properly. The main functional groups present in the original AA side chain are an amine group, a carbonyl group, and a hydroxyl group. The original side chain contains these 3 chains that are twisted around one another, forming a triple helix. Each one of these chains is surrounded by a sphere that allows hydrogen bonding in the network to be present. The hydrogen bonding only occurs when the amine group, in form of a glycine, forms a peptide bond with the carbonyl of an adjacent residue. The hydroxide group comes from one of the amino acids present in type 1 collagen, which is hydroxyproline, which is a derivative of the proline, but an extra hydroxide group is attached to the amino acid. The original AA side chain of the type 1 collagen includes 3 amino acids, which are proline, glycine, and hydroxyproline. The original AA side chain is hydrophobic and therefore does not attract water. This is due to the fact that proline, a side chain that is found in the type 1 collagen, is hydrophobic. The size of the AA side chain can be considered to be smaller than most bulkier amino acids because glycine is considered one of the smallest amino acids and proline is also one of the small ones that we know today. Proline's hydrophobic side chain will repel any water and only associated with other varying hydrophobic side chains. However, in contrast, the glycine is a neutral amino acid that can interact with hydrophobic and hydrophilic environments due to the small nature of the hydrogen side chain on it. But because proline and glycine are considered nonpolar, they are unlikely to form bonds with other charged molecules. Now, as for the new mutated AA side chain of the type 1 collagen, includes the amino acids which are proline, hydroxyproline, and cysteine instead of glycine this time. Similar to the original AA side chain, proline's hydrophobic side will also repel water and only associate with varying hydrophobic side chains. However, in this new mutated AA side chain consists of the following functional groups. You have carboxylic acid, a amino, a sulfurhydryl group, and a hydroxide group. However, as to size, this one is also very small considering the amino acids that make it up. Because the mutated AA side chain is polar, it can participate in forming hydrogen bonds as protein donors or acceptors, which the original one cannot do. This one is nonpolar. The dominant negative substitution of glycine for a larger amino acid in the several locations causes abnormal type 1 collagen cross-linking. Essentially, the lack of normal type 1 collagen can lead to insufficient production of substance in the original matrix of the bone. Essentially, this leads to disease that results in dramatic weakening in the structure of the bone. The models that I have here today presented may be limited because they do not have the amino acids or functional groups that make up each of the original and mutated AA side chains in the type 1 collagen protein. Thank you.